Good afternoon, everyone. This is Daniel Auclair, Senior VP of Research at the Multiple Myeloma Research Foundation, and we're very excited once again to be partnering on this year Myeloma 2017, truly one of the most interesting scientific research myeloma event of the year. So I'm here today with four great panelists. First, Dr. Irene Ghobrial from Dana-Farber Cancer Institute, then Jonathan Keats from the Translational Genomics Institute, TGen, Nina Shah from UCSF, and Dr. Larry Boise from Emory. So let me start with you, Irene. For the first time this year, we had a full session on blood biopsies, an emerging field, and you presented some of your very groundbreaking, exciting data on what you've been doing. Tell us a little bit about these blood biopsies that are emerging and what they mean for our patients.
Yeah, absolutely. Thank you, Danielle. And it's really an exciting time for us. For the first time, we are talking truly about blood biopsies, and in general, can we replace bone marrow biopsies for our patients? Again, many of our patients, I think almost all of the patients will say they do not like bone marrow biopsies. But the beauty of it is not only because it's easier to do than a bone marrow biopsy, it actually gives you a lot of information about the clonal heterogeneity of the disease, about the tumor fraction truly, and what is going on in the changes in mutations, in copy number alterations. And you can detect all of that in the blood versus only in the bone marrow biopsy. But the beauty of it too is that you can get then sequential samples and track those clones as you go on with treatment, whether it's response, resistance to therapy. So there's a lot to be done. It's not only about the DNA. Hopefully in the future we'll get also about RNA, epigenetics, methylation, you name it. So hopefully we will have more and more of the blood biopsies in the future.
Yeah, very exciting. Next to you, Jonathan, you're our scientific lead on our MMRF COMPASS study. We heard a lot about it during this meeting. You're the brain behind all that great genomics that was done on COMPASS. And tell us a bit what you've seen from a genomic perspective during this meeting.
Well, there's been a number of really impressive presentations. I think some of the stuff on the blood biopsies was groundbreaking to see. We've seen stuff about CAR T cells probably not caring about the genomics, which was a huge change. And then it's amazing to see some of the work that was even presented on single cells, trying to get a good understanding of how the tumor and rare populations that may be really driving some of the relapses. From the COMPASS side, we presented some data on novel subtypes of the disease that I think have been underrepresented, that we didn't really appreciate existed. So we had a group of patients that look like the group of patients we would think normally have a translocation, but they don't have a translocation, but they have a lot of poor prognostic markers. And then even something where a number of companies have been working up therapeutics. From the COMPASS study, we really highlight a gene called NIC as being really commonly hit by the tumor. And we know there's other mutations upstream of that gene that having a therapeutic at that position could be very beneficial for a large number of patients. So I think from a lot of the things presented and what we presented, there's a lot of really novel stuff that's been presented at the meeting this year. It's been excellent.
Indeed, indeed. So Nina, you represent along with Irene here, the next generation of multiple myeloma trialists. And of course, being at UCSF, one of the Parker Institute centers, I'm sure immune is close to your heart. What have you heard from a clinical trial perspective during this meeting?
Yeah, it's been really exciting because we had not only one, but two sessions on immunotherapy here. And I would think maybe 4 years ago, we wouldn't have even had one talk. And these sessions highlighted some of the interesting things that are happening with immunotherapy, sort of in four categories. For example, antibody therapy with things like daratumumab, and how daratumumab works not only to kill cells, but change the immune system also. We also heard about the exciting data that's been coming out with the chimeric antigen receptor T cells, or CAR T cells, and we're all very excited about that data and to take those trials more forward and have them be a little more accessible to patients. We also talked about some new things on the horizon. For example, what we call bispecific T cell engagers, or BiTEs. So this is a way to use an antibody to bring T cells to the actual target myeloma cell. And we had a really nice lecture by David Avigan on vaccines. And we're really hoping that somehow we're going to figure out how to sequence all of this so we can get a better immune therapy product to the patient and sooner in their disease.
Yeah, fantastic. Now to Larry. Larry's our kind of dean of the translational efforts that are at the front line in myeloma. And actually you presented some beautiful work on a new exciting class of agents called these BCL inhibitors. What did you see from a novel target, novel drug perspective?
So, thank you, Daniel. So, yeah, I think it's been a really exciting meeting. I think we've learned a lot, some new targets. I think, actually, I'm very excited about the NIC possibility since it's dysregulation in myeloma moving forward. We also learned a lot about resistance mechanisms to our current therapies. So, some very exciting data from Ghobrial on IMiD resistance, and also new IMiDs that are coming down the pike that are very potent and look very exciting, as well as potential other targets that are involved in the response to IMiDs. So that was very exciting. We also heard about proteasome inhibitors and resistance to proteasome inhibitors as well. And as you mentioned, we talked a little bit about the BCL2 family and the success of venetoclax in a subset of myelomas, the 11-14 translocation myelomas, and that maybe it's not limited to just that, but maybe even more exciting, the prospect of having MCL1 inhibitors in the disease.
Yes, indeed, fantastic. All right, quickly, one quick round here. Beside what we just discussed, what do you think is maybe most exciting and could come to patients sooner rather than later that we heard at this meeting today? Who wants to go first?
Sure, I can start with that. I really enjoyed Dr. Ken Shane's talk from the Moffitt Cancer Center on using both in vitro patient samples and mathematical modeling to predict responses to not only single therapeutics but to combination therapies, and how he's doing this in a way that he can actually present a report to the clinician on how their models will predict how a patient would respond to multiple different diseases, and I thought that was different therapies, and I thought that was really exciting.
Nina?
Yeah, I really liked that lecture also with in silico modeling. And I think something else exciting that we talked about, which we've been debating for a long time, has been minimal residual disease. And we had a very heated panel on this because, as people have mentioned before, it's a great thing to try to achieve MRD negativity, but we aren't sure how to get there, and we're not sure what to do once we get there. So I think trying to get intelligent trial designs, which actually the Europeans are ahead of us there, trying to use MRD negativity as a starting point for clinical decision-making is going to be something that our patients, I think, are going to be very happy about eventually. So I'm excited to get that more consistent across centers and for us to really consider it as an endpoint for drug development.
Jonathan?
I'd follow up with Nina that the MRD stuff I think was some of the most impressive, especially from my side. I don't deal with patients all the time, but the most common question I get when I meet somebody is, I've been on maintenance therapy for X number of years and I'm in a complete CR, so when do I get to stop taking therapy? And to see that we actually talked about trials that are activating or soon to activate that are going to start to actually test, can we start taking people off who are in MRD negativity for X amount of time? I think for the patients that I actually interact with from that perspective, 5 years from now, I look forward to not hearing that question or being able to say, yes, you should stay on maintenance, because it'll be great to actually have data to say what people should and shouldn't do, because today we just don't know.
Absolutely.
It's hard to choose. We had so many great things. I think the plasticity work with RNA sequencing yesterday was very exciting because for the first time again, we're trying to tackle that it's not just the CD138 plasma cells that cause all of this, and going beyond what is truly the initiation event that happens and when does it happen, in an early B cell, in a stem cell or not, was very interesting. I think the functional genomics that was presented today with Constantine and adding to it drug screens could be very powerful to detect new drugs, new developments, and potentially even mechanisms of resistance. So it is an exciting time again in myeloma, and it always will be, hopefully.
Absolutely, such an exciting time. And we hope to be back here next year and present even more interesting result, maybe even closer to the patient. So thank you, all of you guys, for your time, and looking forward to see the great progress we all make together for our patient in the next few months and years. Thank you.